Hello, everybody. Welcome back to Virology. Today, we're going to finish talking about how virus-infected cells produce viral nucleic acids. We've talked about RNA viruses. We've talked about DNA viruses. And now we're going to talk about RNA viruses where there is a DNA intermediate. And we'll talk about reverse transcription, the process of making DNA from RNA, and integration. And maybe you've noticed in my lectures I have a quote on the first slide. This is one of my favorites from Alice in Wonderland. One can't believe impossible things, said Alice. I dare say you haven't had much practice, said the Queen. Why, sometimes I've believed as many as 6 impossible things before breakfast. Kind of encapsulates today's subject. Retroviruses were once called tumor viruses. In 1908 and 1911, 2 separate discoveries of viruses that could cause cancers. First, the discovery in 1908 of chicken leukemia virus, and 1911, the discovery of Rous sarcoma virus. And we will talk about these later when we talk about virus-induced cancers. So they were initially called tumor viruses, and then many years later were found to have an RNA genome, and then they were called RNA tumor viruses, which is what they're called to this day. An early investigator in the field was Howard Temin. We'll talk more about him later as well, but he noticed that these RNA tumor viruses, when you infected cells with them, they caused permanent changes which are collectively called transformation. And along with the transformation, viral DNA was integrated into the genome of the host cell. It became a permanent part of host DNA. And so he came up with the provirus hypothesis, which meant, or which suggested, that the viral RNA, in order to make the cells have a permanent new property, transformation, he thought, well, the RNA can't do that. It has to be converted to DNA. And he said it has to integrate into the cell, and he called that a provirus. David Baltimore, at the same time, had been working on the enzymes that copy RNA genomes. And he had noticed, and he, of course, made the Baltimore scheme based on these observations, that plus-strand RNA viruses, there's no RNA polymerase in the particle, remember, because the RNA is plus-stranded, all the proteins can be made when it gets into the cell. For minus-strand RNA viruses, they must carry an RNA polymerase in the particle because the minus strand of RNA cannot be translated in the cell. It has to be copied to make mRNAs first. And so building on Temin's provirus hypothesis, he said, if there is an enzyme that copies RNA to DNA in these RNA tumor viruses, it must be in the virus particle. Because at the time, the thinking was there are no enzymes in the cell that can copy RNA into DNA. We'll see in a moment that this is not correct. So the two of them independently discovered reverse transcriptase in RNA tumor viruses. And they both published these papers independently in Nature. Here's Temin's RNA-dependent DNA polymerase in virions of Rous sarcoma virus. And he noted here this supports the provirus hypothesis. And then for Baltimore, RNA-dependent DNA polymerase in virions of RNA tumor viruses. Now, Temin has since passed away, but David Baltimore is still living. And last year, I did a TWIV with him and some other people who were around at the time of the announcement of these discoveries. And I'll put the link here on the page because in it, Baltimore remarks that Temin had been working on this hypothesis for 10 years. And then Baltimore, in 2 weeks, he had this idea that the enzyme should be in the particle. And he did the experiment. He was getting ready to publish it. And he called Temin and he said, I'm going to publish this. And then Temin rushed his into press. And they both got their articles in the same journal issue, which I think is really nice. The enzyme that copies RNA to DNA is called reverse transcriptase. In fact, retroviruses got their name from their ability to reverse the flow of genetic information. Normally, we think of DNA flowing to RNA, flowing to protein, but of course, reverse transcriptase copies RNA to DNA. Reverse transcriptase has revolutionized molecular biology. For example, the SARS-CoV-2 PCR assays all utilize RT-PCR. Many of them utilize RT-PCR. And you can buy reverse transcriptase and use it to clone genes and to do diagnostic assays. It's been an enormous advantage to doing all kinds of science. So not only did the discovery advance our understanding of how retroviruses work, but it has advanced other fields as well. So today's cast of characters in the pattern of the Baltimore scheme are two viruses or two groups where the viruses have reverse transcriptase, RT, in their reproduction cycles. We have retroviruses where the particles package plus stranded RNA, and this is converted to DNA, first single-stranded and then double-stranded, which again then, of course, can be transcribed to make mRNA. The other virus family we're going to talk about are the Hepatnaviruses, hepatitis B virus, where the DNA is gapped. And we'll see this virus, although you can't tell from this slide, this virus also has reverse transcriptase in its reproduction cycle. So technically, hepatitis B virus is a DNA virus because it's DNA that's packaged in the virus particle. So let's take a look at a typical retrovirus. This is Rous sarcoma virus, one of the first two discovered in 1911. It's an envelope virus with spikes in the envelope that mediate attachment to host cells. Within the envelope is a capsid or nucleocapsid, which contains 2 copies of the viral plus stranded RNA. There are also molecules of reverse transcriptase in the particle. There are molecules of integrase and protease, and the function of these will become evident today and on Wednesday. Let's take a look at the genome of some of these retroviruses. We divide them into 2 categories: retroviruses with simple genomes and with complex genomes. So here, a retrovirus with a simple genome, which means that it encodes fewer proteins than the complex genomes. So here on the top is a diagram of the viral DNA as it would be integrated into the host DNA. We'll see how this happens in a moment. But the DNA encodes 3 major regions of proteins: the GAG proteins, which are structural proteins, the POL, which encodes the reverse transcriptase and the integrase, and then the envelope region, which encodes the envelope glycoprotein. And the structural proteins are further processed into smaller proteins like the capsid protein, which builds the nucleocapsid. Now, that double-stranded DNA is also flanked by 2 sequences called long terminal repeats, or LTRs. Those are viral sequences. We'll talk about those in a moment. This DNA is transcribed in the host genome to produce a long mRNA, the viral mRNA, which codes for GAG, POL, and envelope. Translation by ribosomes produces a GAG, POL precursor. There is a stop codon right after the POL gene, and that is further processed to give rise to structural proteins and the enzymes. The RNA is also spliced to remove the GAG and POL sequences to give an envelope mRNA, which gives rise to the transmembrane glycoproteins. The way these viruses infect cells, they bind receptors on the cell surface. Some of them are taken up by endocytosis. Others fuse right at the cell surface. In this case, the nucleocapsid ends up in the cytoplasm, and there reverse transcription occurs. A double-stranded DNA is made. This DNA enters the nucleus, where it integrates into host cell DNA. And we're showing the viral DNA as blue here, and the host cell DNA is purple. It's transcribed to give rise to messenger RNAs, which are eventually transported to the cytoplasm, give rise to structural proteins that eventually lead to the formation of new virus particles. and we'll talk about that in some detail next time. The glycoproteins are also made by mRNAs exported from the nucleus into the cytoplasm and translated on the endoplasmic reticulum. And those glycoproteins are transported to the plasma membrane by the vesicular transport pathway and eventually incorporated into virus particles. So reverse transcriptase, the enzyme, requires a primer. So here we're showing again our template primer combination. The primer for RT can be DNA or RNA. The template can be RNA or DNA, but only DNA is made. Only dNTPs, not rNTPs, are incorporated by the enzyme. So the primer and the template is flexible, but in the end, the only thing that's made is DNA. Now, reverse transcriptase, it turns out, is not just found in retroviruses. After its discovery in retroviruses, it was subsequently found throughout cells. And in fact, both bacteria, archaea, and eukaryotes all have cellular RT activity. So here we have a phylogenetic tree of life. Just to put this in perspective, we have our bacteria, our archaea, and our eukaryotes, and they all arose from a common ancestor. Therefore, because all three of these have RT must have evolved before the separation of the three. It's an ancient enzyme. And we think that reverse transcriptase is the bridge between the RNA world, which existed early in the life of the Earth, and the DNA world, which came later. So it's sometime in the RNA world, reverse transcriptase arose. And bingo, you could make DNA, and then DNA became the genetic information of the modern DNA world. It can get much bigger than RNA, so it can give rise to complex cells and organisms. Reverse transcriptase, besides being present in bacteria, archaea, and eukaryotes, is also found in hepatitis B virus, as we will talk about today. It's also found in a family of plant viruses, the Colimoviridae. On the lower right here is a circular phylogenetic tree of all the different kinds of RTs that are found. And so in blue here are the virus or virus-like RTs. We have the Hepatnaviruses, the Retroviruses, the Colimovirises, and then there are some movable elements. They move about the genome by virtue of reverse transcription. They are virus-like RTs. We have RTs in bacteria and archaea, and we have RTs in the telomeres, the ends of chromosomes. We'll talk about that later as well. So many, many different kinds of RT. And what I find interesting is that Baltimore looked in retrovirus particles because he figured the cell doesn't have RT, so it must be in the particle. If he knew the cell had RT, maybe he wouldn't have looked. Who knows? Now, RT is very much like the other three kinds of nucleic acid polymerase. Here is HIV, RT. It looks like a right hand with thumb and fingers, subdomains. The thumb and fingers do not touch here in the RT. There is an active site in the palm, which is shown here in the yellow and the red beta strands. And the RT has a triad of amino acids, conserved amino acids in the active site. For HIV1, it's MET, ASP, ASP. And as you might guess, these are coordinating magnesium metals during the catalysis. But HIV-RT, very similar in overall structure to the other three kinds of nucleic acid polymerase, and also with conserved regions. RT, reverse transcriptase, does not just copy RNA to DNA. It has a second enzymatic activity called RNase H. RNase H cleaves RNA when it's in duplex form, double-stranded. So it could be RNA-RNA or RNA-DNA duplexes. It will cut either one. It makes what are called endonucleolytic cleavages. That is, it cleaves internally. It doesn't chew in from the ends. That would be an exonuclease. Endonucleolytic cleavages, and it makes short pieces of RNA oligonucleotides with a 5' phosphate and a 3' hydroxyl. It basically chews up the RNA, and you will see why this activity is needed during reverse transcription. Basically, we have to get rid of the RNA template once we make a DNA copy. Here's the three-dimensional structure of the reverse transcriptase of HIV-1. This is a protein made up of two different subunits, a P66 and a P51, that come together to form the enzyme with the typical palm and fingers and thumb subdomains. And of course, the palm contains the active site, and we see here a molecule of RNA in green has passed through the active site, and a DNA copy has been made. So we have an RNA-DNA duplex here. And then a little bit further away from the active site of the RT is the RNAH site. So it's separated from the catalytic site. And the RNAH, as this duplex passes, it's going to chew away the RNA strand. Reverse transcription is slow. Takes about 4 hours per genome to copy one genome. And it makes a lot of mistakes. It makes a mistake every 10,000 to million bases. And on the left is a cartoon of reverse transcription. We have RNA coming in on the left here, single-stranded RNA. It passes through the active site. There are two metals, and their DNA is added based on the template, of course, by incorporation of DNMPs. We now have double-stranded DNA RNA passing across the enzyme. It reaches the RNAH active site. The RNA now is degraded, and out comes a single-stranded DNA, which eventually will be made double-stranded before it integrates into the host cell. So our first question is, reverse transcriptase has revolutionized molecular biology. Which statement about the enzyme is wrong? It is unique to retroviruses. It is packaged in the particle. It also has RNAH activity. The name comes from its ability to reverse the flow of genetic information. It might have bridged the ancient RNA world and the DNA world, which is wrong. Of course, what is wrong is that, it is unique to retroviruses. We now know it's found in bacteria, in archaea, in eukaryotes, and other viruses as well. Everything else is correct. So let's talk about how reverse transcription works. First, the template. Here's the virus particle on the right here. Each virus particle packages 2 copies of the viral plus-stranded RNA. They're 2 identical copies. And they are held together at one end by what are called kissing interactions. So if we look up there at the left end, there are a series of stem loop structures here. And this particular one, stem loop 1, will base pair with stem loop 1 from another RNA molecule. So here are the 2 RNA molecules. These loops are complementary, inverted complementarity. So they form this kissing loop, which holds the 2 RNAs together when they're being packaged. Also of importance here is this red area on the 5' end of the RNA, labeled PBS. This is the primer binding site. And the primer for reverse transcription is a molecule of tRNA, which is bound to the PBS. You can see it up here in panel A. So every retrovirus particle comes in with 2 copies of viral RNA and 2 tRNAs that are hybridized to the viral RNA, and they're going to serve to initiate reverse transcription. In addition, of course, the RNA is coated with the nucleocapsid protein. And it's not naked, which is one of these exceptions for these plus-strand RNA viruses. I always tell you, most of these plus-strand RNAs are naked in the virus particle. Well, not all of them. This is one that is not naked. You know which one is the other that's not naked? The plus-strand RNA virus, the coronaviruses. They're coated with nucleocapsid protein. And there are about 50 to 100 molecules of reverse transcriptase per virus particle. So we only showed 2 in these drawings, but there are many more. So why are there 2 copies of the RNA, which we call an RNA dimer? We think this makes the viruses relatively resistant to mutagenesis caused by ultraviolet light or ionizing radiation. because they have two copies of all the genes. They have two genomes. So during reverse transcription, what actually happens is that the reverse transcriptase goes back and forth between the two strands, just randomly flicks back and forth. A little bit of DNA is made from one, then another, then it go back and forth, and you end up with basically a hybrid or mosaic DNA that's a bit of each of the two RNAs. And so we think if there are any mutations in the one of the genomes, for example, let's say there's a mutation in gene A. The red is a mutant form of gene A. Well, just randomly, the enzyme will copy the wild type form of A. And so that mutation's effects will be alleviated. So this is called recombination. It's called copy choice because the polymerase goes back and forth between the two RNAs. So it essentially builds one functional genome from two that may be mutated. All right, so in the virus particle, we have two copies of the RNA, and here's the very 5' end of one of those two RNAs, and there's some important parts here. The primer binding site is shown, and the tRNA is shown hybridized to it. tRNAs, of course, have a secondary structure, as shown here, and they base pair. So there are bases on the tRNA. They're complementary to the PBS on the viral RNA. The tRNA inserts itself into the viral RNA as shown here. And there's a free 3' end that's going to be the primer for reverse transcription. This is near the 5' end of the RNA. You can see there's a 5' cap there. And then the rest of the genome is to the right. There are also some important repeated sequences that we need to note here. On the very 5' end, there's a sequence small r, and then another one called small u5. And we're going to see what they do in a moment. Okay, so the virus nucleocapsid is deposited into the cytoplasm, and there reverse transcription begins. Here's the viral RNA. We're showing it circularized, and you'll see why in a moment. The reverse transcriptase uses the 3' end of the tRNA as a primer and begins to synthesize DNA. The blue is the DNA here. And as it's synthesizing the DNA, it's chopping up the RNA. The RNase H activity of RT is chopping up the RNA. So because the RNA is plus stranded, that first strand of DNA is minus stranded. It's actually the wrong color here. It should be a light blue. I have to fix that in the next edition. But look at what happens here. After a short stretch of making DNA, you reach the end of the RNA, the 5' end. Why would you do that? That doesn't make any sense. How can you copy the whole genome? Well, you'll see why in the end, why it does this. But what happens is the enzyme then switches to the 3' end of the genome, which happens to be the exact same sequence as the 5' end, little r, see? So the 5' end, remember I told you, was little r u5. The 3' end is little r u3, little u3. So this little r, these two little r's are the same. The big R is complementary to the little r, and so that DNA that's made can base pair with little r and allow the reverse transcriptase to continue copying from the 3' end of the RNA. That piece of DNA, that short piece of DNA that's made is called minus strong stop DNA. And in order for the rest of the genome to be copied, that big R base pairs with the little r, and then the enzyme continues around the circle, which is shown here in the next slide. So here is what's called the first template exchange. The polymerase is going from the 5' end of the RNA. It's now switching to the 3' end. We have capital R, little r annealing. And the enzyme continues around the circle. And as it's continuing, it chops up the RNA and eventually gets all the way to the tRNA. It does not chop up this short sequence of RNA called the PPT, the polypurine tract. And that's because this is going to serve as the primer for plus strand DNA synthesis. And so even before minus strand DNA synthesis is done, plus strand DNA synthesis begins at the PPT. So let's take it to the next step. Here we have completion of minus strand DNA synthesis. We have initiation of plus strand synthesis from the PPT. So that copies the U3, the R, and the U5, and copies the PBS sequence from the tRNA primer. So this sequence, PBS sequence, the primer binding site sequence, is copied once from the RNA genome here and once from the tRNA primer. Remember, this PBS is actually copied here. As this DNA comes around, it will displace the tRNA and copy the PBS. And then the other strand, the plus strand, is copied from the tRNA at the PBS. The RNA primers are removed. The RNAH removes this primer, and it also removes the tRNA. And then the enzyme can continue filling in the rest of that plus strand. So this is called plus strong stop DNA. It can't go any farther than the tRNA until it anneals to the PBS on the first DNA strand that was made. Those are complementary. And so now these DNA ends are juxtaposed by annealing, and the enzyme can go around. So that is the second template exchange, essentially. The enzyme, let me go back, is going from copying this strand here. It can't go any further. And then it switches to the other end of the genome here. And that's the second template exchange. So here we now have a fully single-stranded DNA and a partially double-stranded DNA. The enzyme continues to make plus-strand DNA. It's going all the way through until we have a complete double-stranded DNA. And I want to point out now what has happened. We now have a DNA with U3, R, and U5, capital letters, U3, R, U5, U3, R, U5. So we have two copies of this sequence, and that's the LTR, the long terminal repeat. There's one at each end. We also have a PBS and a PPT, which are the primer binding sites, of course. All right, so let's take a look at this in some detail. So here's the viral RNA, which we start out with. At the 5' end, there's little r, little U5. The tRNA is annealed. After copying, we end up with two LTRs, each of which has capital U3, capital R, capital U5. So this weird kind of synthesis where we have two template exchanges, one here and one later on, that is needed to build two LTRs from these sequences at the ends of the RNA, at the 5' and 3' ends of the RNA. Because the RNA does not have complete LTRs at both ends. Why that is, you'll see in a moment. So it has to be rebuilt. And the rebuilding is done by this weird two-template jump kind of synthesis. So with a short genome RNA, you end up duplicating the ends and making two LTRs. And if you don't quite get this reverse transcription, I made an animation some years ago for this class, and you can find that there at YouTube. Our next question is, which of the following steps occur during reverse transcription of retroviral genomic RNA? Priming of minus DNA synthesis by tRNA, two template exchanges, degradation of the viral RNA by RNAH, generation of two LTRs. Which of the following steps occur, or all of the above? The answer is all of the above. All of these are right. Priming of minus DNA by tRNA, two template exchanges, degradation of the RNA by RNAH, generation of two LTRs. They're all correct. So what we have done is to make a double-stranded DNA copy of the retrovirus genome, which has at both ends a copy of the LTR, the long terminal repeat. What's going to happen next is that this DNA is going to integrate into the host cell DNA, and then we will end up with what is called proviral DNA. The proviral DNA, the definition is an integrated copy of the viral DNA. That's what we mean by provirus. So we have an LTR here on the left, the coding sequences of the viral genome, and an LTR on the right, and it's both flanked by host DNA. And two very important things happen during the integration of retroviral DNA into host cell DNA. First, some bases at the ends of the LTR are lost. In this case, shown about two bases are lost. These two A's are missing, so you can see we just have TG here. Same happens at the other end. The second is that host DNA sequences at the ends are duplicated. And so this red sequence and this orange sequence are exactly the same. So they're not present before the integration. We'll see why these things happen in a moment. And this proviral DNA is then the substrate for transcription, the synthesis of mRNA. There is a promoter, a strong promoter built into the LTR. So both LTRs have a strong promoter. And the left-hand promoter gives rise to the transcription of the viral RNA, which is capped and polyadenylated. And this can then exit the nucleus and give rise to viral proteins, or it can also be spliced and exit the nucleus. So let's take a look at the integration reaction. This is an activity of another viral enzyme, the integrase. So again, the viral nucleocapsid comes into the cytosol, reverse transcription occurs in the cytoplasm, and then this complex of viral double-stranded DNA enters the nucleus. And there, the integrase, which is this kind of donut-shaped, brownish protein there, that's the integrase. That's going to interact with cell proteins that are bind to the host cell chromatin. So here's the nucleus of the cell. Here's our chromatinized DNA. BRDs are bromo domain containing proteins. These bind acetylated lysines on histones. And the integrase binds them. That brings the viral DNA in proximity to host cell DNA, and it initiates the integration reaction. So again, the DNA is made in the cytoplasm. It's imported in the nucleus, where it then can bind chromatin via the integrase. This protein BAF, it stands for barrier to autointegration. It's thought to prevent the DNA from integrating into itself, because obviously that could happen here, right? The integrase could just integrate into its DNA into DNA, which would be non-productive. So this protein apparently prevents that. How does integration work? Here's a schematic of it. The integrase, IN4, is a tetramer. So we have retroviral DNA produced by reverse transcription now. We have the two ends, which are 5' and 3' ends, nice flush ends. And the first thing that happens is that the integrase removes two bases from the 3' ends of both strands. So that's an enzymatic reaction. Two bases are taken off, and now you know why two bases are missing from the end. And the reason you do that is to make a hydroxyl that can attack the target DNA. So these 3' hydroxyls now can attack the target DNA, can nick it and be ligated to it. And because of the way that this is happening, you can see the two strands of the host DNA, the light purple and the deep purple. They're going to be eventually copied in, repaired, and ligated. So that sequence is going to be duplicated, right? Because you're copying the top and the bottom strand. So the host DNA is ligated to the viral DNA. The gaps are repaired and ligated. And now we have integrated or proviral DNA. And the host sequence at either end has been duplicated. The orange and the red are duplicated sequences. So this integration can occur at many sites on both strands of DNA, but it's not random, and it really depends on the particular retrovirus, what the pattern is. And here's just a study where the percent integration of 3 different retroviruses is being shown by examining integration sites in infected cell DNA. And this is an avian retrovirus, a murine retrovirus, and HIV. And if this integration occurred at random, you would expect 26% integration within genes, 5% within transcription start sites, just based on the number of these sites. For the avian retrovirus, it's 42% and 8%. For MLV, it's 40 and 20. For HIV, it's 60 to 70 and 10. So it's not random. The numbers do not match what you would expect from randomness. And so this could go these viruses can integrate almost anywhere in the genome. So to summarize, we have a single DNA that's been produced from 2 separate RNAs now, remember, by reverse transcription. In the particle, there are 2 RNAs, but those 2 RNAs give rise only to 1 DNA. In the LTR, a strong promoter is built during reverse transcription. It's built because the RNA doesn't have the complete promoter sequence in it. The proviral DNA, the integrated viral DNA, directs host transcription. There's a promoter there in the left and right LTR. The left-hand promoter gives rise to transcription of the viral genome. And so you make many copies of viral mRNA, which are then translated to proteins in the cytosol. That RNA can also be encapsulated into virus particles besides being made into protein. So think about this. There's no viral DNA replication here, right? We're making a single DNA from 2 RNAs. There's no viral RNA replication. We are making the new genomes by transcription. So it's very unique in terms of the reproduction of the nucleic acids of the virus. So to summarize this, big picture, 35,000 feet, the virus binds the cell, it enters at the plasma membrane, the RNA in the nucleocapsid is reverse transcribed, the double-stranded DNA still in a subviral complex goes into the nucleus, integrates into host cell DNA, becomes the provirus. That is transcribed to form mRNAs that are exported to produce various proteins needed for assembly of new virus particles. And this assembly process is very interesting. We're going to talk about that next time. These proviruses are really interesting and really important. They are permanent. If the cell that's infected doesn't die, the provirus stays there for as long as the cell does, because there's no way for it to get out. There's no way to excise precisely this provirus. The only way out is by transcription. You make an mRNA, and that RNA can then go out and make new virus particles. And if this provirus happens in a germ cell, then it can stay with the species for many, 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 many years, perhaps millions of years. So when the provirus happens in a germ cell, we call this endogenization. And our genomes are full of ancient and modern proviruses, which we call retroelements, and I'll explain why we call them that in a moment. Our genomes, every genome of every living thing on the planet is full. I shouldn't say every living thing. There are probably exceptions. Many, many genomes of cells and organisms are littered with retroelements as a consequence of infection by a retrovirus. And what happens to these is shown at the bottom here. So let's say we have a population of individuals many, many years ago. One of them is infected with a retrovirus, and it infects their germ cells, sperm or eggs. And so now when that person has offspring, the offspring are going to have the provirus as well. And with time, those proviruses, of course, sustain mutations, which we call polymorphisms. Those are the different colored individuals here. And eventually, the provirus may remain in the genome forever. It may become fixed. In some of these people, it may be lost, just randomly lost because it's not needed. And so we can see the evidence of this to this day. For example, In koalas, we can see this endogenization happening right now because this is a very recent event. In humans, the events that led to endogenization happened many, many hundreds of thousands of years ago. But in koalas, about 50,000 years ago, apparently a retrovirus crossed from rodents to koalas. And we know this because we have phylogenetic calculations that tell us this, but also many museums collect koala pelts, and you can go back and show that in pelts hundreds of years old, there's already these retroviruses in them. And this map shows Australia, different locations in Australia, and the pie charts show you the fraction of the koala population that has already been endogenized. And you can see towards the north inland here, very high numbers. And on some islands where it's harder for the virus to spread, there's just a small amount of endogenization. But so this is happening in real time, and it can be studied. I mean, this virus will infect koalas, and so it can be spread by virus infection as well as by giving birth to offspring. So this is real-time endogenization, and we can study it. So I've used this term retroelements a few times already. Let me define what I mean by that. A retroelement is nothing more than a sequence that moves around the genome by reverse transcription. Here's an example of that. We have a sequence here integrated into the genome. It's transcribed at some point to make an RNA. And in the nucleus, there is also reverse transcriptase. A lot of genes in our genome encode reverse transcriptase. So this RNA can be converted to DNA. I'm sorry that the colors are wrong. This is a figure from another text or paper, and they don't use our color convention. They're using red for RNA and green for DNA. Anyway, the RNA can be converted to DNA by reverse transcription, and the DNA can integrate somewhere else. So the net result here is that this gene is now moved somewhere else, and it's been duplicated. And this happens all the time in genomes. Genes are duplicated hundreds and thousands of times. They move all over the place, and sometimes they screw things up. If they integrate into an essential gene, that's a problem. Anyway, this retroelement can be proviral DNA. Those can move around for sure, or it doesn't have to be a provirus, as you'll see in a moment. But if proviral DNA is integrated into our germline, we call this an endogenous retrovirus or an ERV, endogenous retrovirus, because they're endogenized, which means they stay with you as you give birth to kids. The kids have your proviral DNAs. In many species, these are often replication defective, these ERVs. In humans, they're all defective. They don't make infectious viruses. They can make particles. The integrated proviruses can make particles, but they don't make infectious particles. And look at this. 42% of the human genome is made up of mobile genetic elements, sequences that move around, in many cases by reverse transcription, including these endogenous proviruses or retroviruses and other retroelements as well. We're going to take a look at this right now. All right, this is a table and a picture showing you all the retroelements in the human genome. Again, a retroelement, just hang on the word retro. It means it moves around the genome by reverse transcription. 42% of our genome is retroelements. 2.7 million individual retroelements, and they're broken down into different kinds. So let's take a look at the picture here first. First, these are all retroelements. They move around the genome by reverse transcription. We have endogenous retroviruses. These are the consequence of infection many years before by a retrovirus which has gone into the germline. So it can be passed to your offspring. So we have host DNA duplicated on either side of the provirus. We have 2 LTRs, and we have gag pollen envelope. It's just a classic retrovirus, right? 2 LTRs and 2 duplicated host sequences. So you know this went in by integrase because of the duplication. Then we have what are called retrotransposons. So these you can see have duplicated host DNA. They have 2 LTRs, they have GAG and POL, but they don't have an envelope. They can't make a particle because there's no envelope gene. You could make a membrane-encased particle, but it wouldn't be able to infect another cell because it doesn't have a spike glycoprotein in it. These were probably the progenitors of retroviruses. They were probably around before retroviruses, and then at some point they captured an envelope gene from the cell and became a virus and now could go from cell to cell. So retrotransposons were in cells for a long time before retroviruses. So far we have, let's look at the chart here. So LTR containing retrotransposons. We've just talked about those two kinds. The endogenous retrovirus is about 7.7% of our genome, and the retrotransposons about 0.6%, okay? And then we move to other non-LTR containing retroelements. We have lines, long interspersed nuclear elements. These have duplicated host DNA on either side, so went in by some kind of integration reaction. And then no LTRs and just a couple of open reading frames. And these don't make infectious particles, but one of these open reading frames encodes reverse transcriptase, and that is how these could move around the genome. Remember, these are all elements that move around the genome and duplicate and triplicate and quadruplicate themselves. So lines, so a lot of these non-LTR elements, 16.9 and 3.2%, two different kinds of lines are present. 16%, over 20% of our genome is this line stuff here. And then we have two others where there is no reverse transcriptase in the element. We have what are called processed pseudogenes, which have duplicated host DNA. And these are essentially just DNA copies of mRNAs that have been captured by reverse transcriptase from maybe a line or a retrotransposon. So a reverse transcriptase provided by a different element. And then signs, small interspersed nuclear elements, again, an open reading frame with no RT duplicated host sequence, probably moves around the genome by RT from another element. And processed pseudogenes less than 1%, and the signs are about 12% or so. So those are the retroelements in the human genome, and every species has its own kind of retroelements, and only about 0.05% of all these are active in the genome today, which means that they're being transcribed and making something. In a couple of situations, organisms have actually used integrated retroviral genomes. We call this exhaptation. When you steal a gene from a virus and use it, it's called exhaptation. And a classic example is the syncytians. Syncytians are the proteins that give rise to the placenta. Here's a diagram of the structure of the placenta. We have our fetal villi and the uterus here. An important part of the placenta is called the syncytiotrophoblasts, this red series of cells here. These are actually all fused cells. See the many nuclei, but there's no cell walls there. They're all fused together. That's an important part of their function. They're a very important barrier between maternal and fetal circulation. And so these fused cells are caused by a retroviral spike glycoprotein. Remember, the retroviral genome encodes an envelope glycoprotein. And this is a membrane protein. It's on the envelope of the virus particle. It engages a receptor, and the virus then fuses with the cell. So the spike protein is fusogenic. Well, if you just had the spike on a cell and the receptor for the spike on another cell, those two cells would fuse. And that's exactly what happens with these syncytiotrophoblasts. They produce an exapted envelope. It used to be a retroviral envelope. It's now called syncytian, and it gives rise to the placenta. And this is something that happened multiple times in mammalian evolution. Here we have two different instances millions of years ago of stealing of a syncytium from two different retroviruses that infected ancestors of these primates here. And so this stealing of a spike glycoprotein gene from a retrovirus enables these animals to have a placenta and give birth to live animals without having a pouch like a marsupial. All right, so the syncytium was stolen from a retrovirus. We used it. Hey, it's something we could use. Let's take it. Here's another example of retroviral influence on human embryonic development. This is a particular endogenous retrovirus called HERV-K, which infected Homo sapiens ancestors about 200,000 years ago. So here's Homo sapiens here, right? So our ancestors here, Neanderthalensis and Rhodesiensis, about 200,000 years ago. So it entered the germline about then. And today they don't produce any infectious virus, but the RNAs, the mRNAs from these endogenous proviruses are produced during embryogenesis, all the way from the 8-cell stage to the epiblast stage. So the oocyte, the zygote, 2-cell, 4-cell, no transcription of HERV-K, but starting at 8-cell in the morula and the epiblast, you see transcription of HERV-K and virus-like particles. And these cause induction of an antiviral protein, so maybe they have some role in protecting the embryo from infection. Another example. And finally, if you've ever liked chicken eggs that are blue, you can buy white chicken eggs. You can buy brown ones. You can also buy blue ones. The farmers breed their chickens to give them blue eggs. They'll find one that randomly gives blue eggs, and then they can breed them to get a stock which reliably produces blue eggs. The reason for this color is that a retrovirus has integrated next to the gene that produces blue pigment in the egg. Of course, this is a gene that's expressed in the uterus. The gene is turned on by the retroviral promoter. Remember, the retrovirus inserts into the DNA. If it inserts next to a pigment gene, the eggs are going to be blue. So without retroviruses, we would be laying eggs, and they would be white. Our next question is, which of the following statements about retroelements is not correct? A, there are many copies in eukaryotic genomes. B, they are currently entering the koala germline. C, those in the human genome produce infectious viruses. And D, they can be beneficial. E, none of the above. Which is not correct? Well, the statement that's not correct is those in the human genome produce infectious viruses. That's not correct. There are many copies in eukaryotic genomes that are currently entering the koala germline. They can be beneficial. We've seen syncytiums derived from them, but the ones in the human genome do not produce infectious viruses. Finally, let's talk about the other virus family with reverse transcriptase in the reproduction cycle, and that is the Hepatnaviruses. These are DNA viruses that encode reverse transcriptase. So these are envelope viruses. The envelope contains the viral glycoprotein. Within the envelope is a nucleocapsid that contains this odd viral DNA, which is gapped. Remember, it's a separate Baltimore class unto itself. And it has a protein stuck on it, and it has a piece of RNA stuck on it. All these things have to be fixed before it can be transcribed. But this genome, it's shown here, gives rise to a number of proteins by transcription and alternative splicing. So what's the story with RT? These viruses bind receptors. The nucleocapsid is delivered to the cytoplasm. Now, this nucleocapsid has partially double-stranded DNA in it. It docks on the nucleus. The DNA enters the nucleus. There, the DNA is repaired by host cell enzymes. The protein is removed. The RNA is removed. It's made fully double-stranded. It's chromatinized, so it's now covalently closed circular DNA. There is no genome integration for these viruses. Hence, there is no integrase encoded in these hepatitis B virus genomes. The viral DNA remains a circular double-stranded DNA. It is transcribed by host cell DNA-dependent RNA polymerase to produce messages that then exit the nucleus and they're translated into proteins in the cytosol. Among the proteins are structural proteins that give rise to building a new capsid. And the genome that is put in that capsid initially is an RNA. It's the pregenome, which is a RNA that's a copy of this entire closed circular DNA. That gets encapsulated into the capsid. But before this particle leaves the cell, it begins to reverse transcribe, and you end up with that gapped, strange, partially double-stranded DNA molecule. So reverse transcription commences in the cytoplasm. That is different from retroviruses, where reverse transcription only happens in the next cell that's infected. For these, reverse transcription begins as soon as that RNA is encapsulated in the particle. Now, why it stops is because the reverse transcriptase runs out of triphosphates. These capsids are not permeable. They are formed and they capture a certain amount of dNTPs. And when those run out, that's it, and you can't make a complete double-stranded DNA. So that's an explanation for why the DNA is partially double-stranded. By the way, eventually, these capsids containing this weird DNA get enveloped intracellularly and then passed on to exit the cell by vesicular transport. So let's take a look at this process of reverse transcription. It actually has some features of retroviral reverse transcription. So here is the viral genome. It's very strange. It's 5' capped. It has a number of secondary structures that are important, like this one near the 5' end. It has a 3' poly A sequence, and it has some direct repeats. So DR1 and DR1 are the same, and DR2 is a different one. So reverse transcriptase is right here bound to the viral RNA. It's bound to this secondary structure. Reverse transcriptase is that beige protein in the back. The primer is a terminal protein linked to DNA. The reverse transcriptase begins to copy this sequence, and then it switches templates to up here. And you can see that AATG is now base pairing with the UUAC on the other strand near the 3' end. So kind of reminiscent of the retroviral template switch. So now the reverse transcriptase begins to copy from the 3' end of the genome there. So again, going from the 5' end, probably this stem loop stops the movement of the enzyme, so it just switches to the other strand, begins to make the other strand until we get now a minus strand DNA. This mRNA, of course, is plus-stranded, so the DNA, the first DNA is minus strand, and here it's the right color. And that DNA has on its 5' end the reverse transcriptase. It's finished and it remains attached to it. The 3' end is then copied using an RNA primer. This is derived from the viral genome. It's simply not removed. But there's a very strange thing that happens here. So the reverse transcriptase goes all the way around, makes the first DNA strand, and then the RNA primer is left. It's not chewed up by RNase-H, and then that serves as a primer for reverse transcription of the second strand. But oddly, it doesn't continue on that same strand. The primer jumps, another template jumps to the 5' end here. You complete the DNA there. The polymerase begins to use that piece of DNA, that piece of RNA, to start priming a second strand synthesis. So we end up with a single long minus strand DNA. The RNA primer is what remains after RNase-H degradation. But that primer is not used for reverse transcription where it sits. There's another template exchange where that primer jumps to the 5' end. The enzyme then copies all the way to the 5' end of that RNA. And then it jumps back to the 3' end of the DNA, where there's a terminal redundancy, and then begins to make the rest of the plus strand. So here there's a little bit of plus strand made from that sequence between the primer and the terminal protein. But then the RT runs into the end of the genome, and then it has to switch to the other, another template exchange. And if all were well, the enzyme would make a full second strand, right? But this is all happening within the particle in the infected cell, and so there aren't enough triphosphates there to complete the synthesis. So here is where synthesis stops. You have a full negative stranded DNA in the light blue, a circle. You can see that here. Actually, it's not a circle. It's got a reverse transcriptase protein bound to the 5' end. You can see it's beige here and it's shown blue here. So that's the 3' end of that negative strand DNA, and it's the 5' end. So it's a complete negative strand, but they're not covalently closed. And then you have a partial plus strand where you have a little bit of the primer left here at the 5' end. You have some plus strand, but certainly it's not a complete copy of the minus strand. And it ends up being like this because this happens inside the capsid where there aren't enough triphosphates to carry out the synthesis that's necessary. So you see a number of similarities with reverse transcription of retroviruses in template exchanges and the use of RNAs derived from the genome as primers. So let's compare now a number of different viruses that have reverse transcriptase in their reproduction cycles. We've talked today about retroviruses, where, by the way, on this picture, the yellow tells you what is packaged in the virus particle. So for retroviruses, of course, most retroviruses package RNA. They package RNA in the form of a dimer with a tRNA primer. When they infect a new cell, this is made into a double-stranded DNA, which eventually becomes a proviral DNA. The proviral DNA is transcribed to form messenger RNAs, which are encapsulated into new virus particles. Now, some retroviruses, called foamy retroviruses, actually package DNA. 5 to 20% of the particles have DNA in them and not RNA. So many of them do have RNA, like retroviruses. But a fraction of these foamy retroviruses package DNA. And again, we think that is a consequence of a similar situation as for the Hepatnaviruses, where initially RNA is packaged, but it undergoes reverse transcription such that the particles that are released have DNA in them. It's weird DNA. It's not fully competent, but it's DNA. And so we think the foamy retroviruses also package RNA initially and then do some reverse transcription before leaving the cell. And that's why you have DNA in a fraction of foamy retrovirus particles. And finally, there are retroviruses of cauliflower mosaic virus, which also have DNA in the virus particle. And again, we think these viruses initially package RNA into the particle, and in the particle where there is reverse transcriptase, of course, before leaving the infected cell, that RNA is copied to form DNA, and therefore the virus particles have DNA in them and not RNA. Now, when retroviruses and foamy retroviruses enter cells, they either put an RNA in, which is copied to DNA, or an RNA or a DNA. In the case of inserting a DNA in, that DNA can directly integrate. Of course, for Hepatnaviruses, it has to be repaired before it can be transcribed. There's no integration, and transcription gives rise to new genomes. Also, for cauliflower mosaic virus, there has to be some repair done before this double-stranded DNA is competent for transcription. There's also no integration for cauliflower mosaic virus. So these are really slightly different variations on the same theme, making an RNA, packaging it, and reverse transcribes either in that initial producing cell or in the next infected cell. This brings us almost to the end of the first half of this course. On Wednesday, we're going to talk about how to build new virus particles. We're going to take all of the genomes and particles that we have made and assemble infectious viruses.